Does anybody have any questions from yesterday before we get into this? All right, no questions? That's fine. Okay, so today we are going to learn about translation. So we've already covered DNA replication, and then we've covered transcription, which is DNA to RNA. And today we're going to cover translation, which is RNA to polypeptides, and eventually to proteins. All right, so we're going to go over a couple of things today. We're going to go over everything that's involved in translation. We're going to go through the mechanisms, and then we're going to learn about something called protein trafficking and post-translational modifications. So translation will only create your polypeptide chain. So to create functional proteins afterwards, you need to go through something called post-translational modifications or protein trafficking, and yeah, we'll get to that. So we know after yesterday's lesson that prokaryotes and eukaryotes have a very different way of going about their transcription. And also now today we'll learn their translation. So already we can just look at their structures, and for prokaryotes, as they create the mRNA, you can see here that their polypeptides are being created at the same time. But in eukaryotes, the mRNA has to first be processed and then it can be brought outside of the nucleus where then it can be translated. So there's a big difference here. Okay, another quick overview. Cool. So normally if you guys were in class, you would have an activity that would basically introduce you to this whole translation idea. Oh, I think my pen's not working. No, interesting. Let's try and reconnect it. Okay, I mean, I guess we're not going to use it today. It's not working. But I can just write. Okay, so normally what you would get is you would get like a little map, and then it would basically be a reference for all the different codon, uh, what is it, codon combinations, and that would refer to specific Lego block colors, and you would proceed to build like a long, basically line of Lego blocks. But we don't have access to that over this call, so we are going to try our best to understand without the genetic Lego block activity. Um, Maybe we can try and answer some colors. No, I don't think this is too important to your understanding. We should be able to get through it without doing that activity. Okay, so let's take a quick look at the slide. Reading the genetic code. So just as we read sentences, our body or our translation mechanisms need to be able to understand the RNA that it has, the mRNA that it has to translate into a polypeptide chain. So it's almost a good analogy here is like in this big mess of words, are you able to make out any sentences? So for the first one, there's like a mess of letters here, doesn't really mean much, but then here you can start from the man was too old for the war end, and then more incomprehensible text. Oops. Or the next one. Again, some incomprehensible letters here. Again, starting with the boy had one red car and one old hat. End. And finally, I won't make the lines for this one. The car, or I'll put the start and the end. The car was red and too old for her new job. End. So you might have noticed a few patterns in these three examples. All of the sentences start with the, and you can think of that as what we'll call from now the start codon. Each word is three letters, and in real life, each codon we'll learn what a codon is in just a second, is 3 bases long, and all your sentences will end with end, and what we'll refer to as the stop codon. We will learn what codons are in just, I think, probably the next few slides. So translation, just like transcription and replication, has an initiation, an elongation, and a termination. Let's go through that. So there are a couple steps to initiating translation. So does anybody know what a ribosome is? So a ribosome is the protein, or I think it's a ribonucleoprotein that will find and bind to the mRNA strand, and the ribosome is basically the protein that will translate your mRNA and create a polypeptide chain. It's very important. The ribosome has two subunits. It has a small subunit and it has a large subunit. And both of them have to be present for the ribosome to be working properly. But they come in at different times. So step one, the ribosome finds and binds to the mRNA strand. So in prokaryotes, the 5' end of the mRNA transcript has a Shine-Dalgarno sequence. So it has a specific sequence that the ribosome will look for and bind to. And the rRNA on ribosomes, so ribosomes are something called ribonucleoproteins, which mean that they are proteins that have their own unique RNA sequence that's part of their structure, important to their function. So there's something called ribosomal RNA on the ribosome, small subunit, which has a complementary anti-Shine-Dalgarno sequence, which helps it find and bind to this 5' end of the mRNA transcript in prokaryotes. In eukaryotes, the ribosome's small subunit recognizes and binds to mRNA at the 5' cap. Very different. Oh, so we'll go over the structure here. Cool. Ribosomes are something called a ribonucleoprotein. So they're composed of ribosomal RNA, which we will refer to as rRNA from now on, and protein. So from the picture, you can see that RNA, which is RNA in a double-stranded helix, is in turquoise, gray, orange, and indigo. So turquoise, gray, orange, indigo. And protein alpha helices are in violet and navy blue. Cool. So, its structure. Ribosomes are made in the nucleolus for eukaryotes. I don't know where they're made in for prokaryotes. I don't know if we ever learned. They have two subunits, like I said earlier. The large one is turquoise and the small one is lime green. Cool. Another basically quick fact. Large is however many units. Small. Okay. So step 2. What happens after the small subunit binds? The ribosome then locates the translation start site. So the ribosome's small subunit moves along the 5' leader of mRNA until it reaches what we call the translation start site, or the start codon, AUG. Afterwards, there are certain factors that will help the ribosome's small subunit, there are factors that will help the ribosome's small subunit find the start codon. In prokaryotes, they have something called initiation factors. And remember earlier we learned about the pre-initiation complex in eukaryotes. This is completely different. So here prokaryotes have initiation factors that help the ribosome small subunit find the start codon. In eukaryotes, there's something called the Kozak sequence on the mRNA. And this is what the ribosome small subunit looks for when it's trying to find the start codon. So what are codons? Because I've been throwing that term out, but I haven't really explained what codons are. So a codon is three consecutive RNA nucleotides, or a triplet, on the mRNA, the transcript that was transcribed from DNA. Each triplet or each codon codes for a specific amino acid. So if we take a look here, you're basically starting with your pre-mRNA and then processing it, you get mRNA. And then if we zoom in, you can see that we're going to group them 3 bases at a time and call them codons. And it's pointing because each codon has its own unique, or I don't know if I should call it unique, but each codon refers to a specific amino acid. So UGG will only ever refer to tryptophan or code for tryptophan. UUU will only ever code for uh, 4 combinations of triplets, so and not that many. Okay, yeah, you'll okay. We'll talk about that in just a second, David. That property is pretty important here. So, well, yeah, I understand that there are 64 combinations of triplets and only 20 amino acids, but there's a there's a reason. And yeah, you're right, but it's perfectly fine, and we'll go over why it's perfectly fine in just a second. So, initiation step 3. The initiator tRNA binds. So once this ribosome small subunit has reached AUG, the start codon, an initiator tRNA will attach to AUG. And the way it attaches is by the initiator tRNA and the mRNA hydrogen bonding. So hydrogen bonds form between the mRNA codon and the tRNA anticodon. tRNAs are called transfer RNA, I think that's what they're called. But tRNA carries the amino acids or carries amino acids to make the polypeptide chain. What are anticodons? Anticodons are complementary sequences on the tRNA. So let's go over the structure of tRNA. So tRNA are basically RNA themselves, but they have a specific structure that allow them to have this anticodon, which will hydrogen bond with the mRNA codon, and also at their 3' end, they have something called an amino acid attachment site. So essentially, they'll like hold an amino acid here, come over to the ribosome, bond to the mRNA, or bond to the specific codon, and then basically pass on their amino acid. And we'll learn how they do that in a second. So, in prokaryotes and eukaryotes, the initiator tRNA is holding a different amino acid. In eukaryotes, the initiator tRNA brings over methionine or MET, but in prokaryotes, it brings over something called formal methionine, FMET. You do have to know this difference, but you just have to know it as far as eukaryotes carry, or the eukaryote start codon codes for methionine or MET, and prokaryotes FMET. Small difference. Okay, step 4. After the initiator tRNA has bonded to your mRNA at the start codon, the ribosome large subunit binds, and basically now you have your ribosome complex ready, and it's ready to go. So we call this entire thing the translation initiation complex, or the complete ribosome, plus the initiator tRNA, plus the mRNA at AUG. Cool. So ribosomes actually have 3 binding sites. One for mRNA, or sorry, they have multiple binding sites. One for mRNA and 3 for tRNA. And the 3 different sites for tRNA have different names. There's a E site, a P site, and an A site. E stands for exit, probably the easiest one to remember. P site is the peptidyl tRNA binding site. I just remember it as the P site. And the A site, which is the amino acyl tRNA binding site. You just have to remember it as like EPA. And for me, I almost just remembered it as like the first site, the second site, and then the exit site. But they have names. So what happens at the different sites? When we first create the translation initiation complex, the initiator tRNA is binded at the P site. But when we actually look at the process of translation, we'll start by looking at the A site, also called the amino acyl tRNA site. This site holds the amino acid tRNA carrying or amino acyl tRNA carrying the next amino acid to be added. So basically this is like where the next tRNA in line holding the amino acid waits. In the P site, this holds the tRNA molecule carrying the growing polypeptide chain. So we'll understand what that means in just a second. And the E site is the exit site, where tRNA molecules leave the ribosome. So what does this mean? Step 1, a codon is recognized. So I guess it like touches down. And step 2, a peptide bond forms over here. So between this amino acid and this amino acid, a peptide bond forms. So as you can see here, between step 2 and step 3, the entire polypeptide chain has been moved from basically the tRNA on the P site to the tRNA on the A site. And then after that, what happens is translocation. So the ribosome moves along the mRNA, and basically all the tRNAs basically move one slot to the left. What happens then? So the tRNA that's on the A site is now in the P site, and the tRNA that's in the P site is now in the E site. And once it's in the E site, it's exiting. So how does this elongation really work? So first, what happens is the incoming amino acyl tRNA comes to the A site. Hydrogen bonds form between the mRNA codon and the tRNA anticodon. Energy is required. And then what happens is a peptide bond forms. So the ribosome catalyzes the formation of a peptide bond between the amino acid in the P site to the amino acid in the A site. And this involves the C terminus, involves the carboxyl end of the polypeptide chain. So I'm pretty sure you guys learned in one of your previous units that polypeptides grow from N terminus towards the C terminus. So the C terminus is growing or being added onto. So what is the result? The polypeptide chain after this step is longer by one amino acid, and the polypeptide chain has been transferred to the tRNA at the A site. And afterwards, the ribosome moves, and like I said earlier, P site to E site, A site to P site, and now this one is ready for the next amino acyl tRNA to come in, and this one can leave. So, ribosomes only move in one direction. The mRNA is bound to the tRNA, and the ribosome translocation happens by one codon, so 3 whole nucleotides. The ribosome reads mRNA in the 5' 3' direction, so mRNA is moved through the ribosome unidirectionally, one direction only, from 5' to 3'. This is another diagram. I think it's the same one as before. So, termination. How does this whole thing end? At the stop codon, so there's a start codon, and then there's a stop codon. At the stop codon, a protein called the release factor, instead of an amino acyl tRNA, binds to the A site. There is no tRNA for the stop codon, and thus no amino acyl tRNA for the stop codon. Instead, the release factor adds a water molecule instead of an amino acid to the polypeptide, and thus the polypeptide is hydrolyzed from the tRNA in the P site and released. So this bond here is broken by water, and the two of them separate. The tRNA is released, the free polypeptide is released, and the entire translation complex disassembles. I have a video for you guys if you'd like to see. I think it's this one. You can start watching it around 112 if you want to see it. If you guys understand, we can just move on. If it's still a bit confusing, we can watch the video together. But I think this part shouldn't be that much to understand. If you guys have any questions, this is the time to ask. Cool. So, polyribosomes. What does this mean? A single strand of mRNA can be used to make many copies of a polypeptide simultaneously. So remember earlier in transcription when we said that as long as the promoter was open, another RNAP can bind? So polyribosomes is a very similar idea, where a single strand of mRNA, as long as there's space for another ribosome to basically initiate and bind, many ribosomes can be translating a single strand of mRNA at the same time. So a polyribosome is when one molecule of mRNA has multiple ribosomes simultaneously translating the mRNA. So this is a diagram, and this is a real life micrograph. So what happens to the RNA after it has been used for translation? To be honest with you, I'm not entirely sure. My best guess is that it's either recycled or maybe kept around. I don't know what happens to RNA after it's been used for translation. Because translation for for mRNA specifically, like translation is basically its only purpose, right? So if it's not needed, then obviously your body will recycle it. That's my best answer. You could ask Miss Law. Okay, so protein, but this polyribosome idea is not really unique to uh, eukaryotes or prokaryotes, to the best of my knowledge. So let's look at protein synthesis in prokaryotes. So prokaryotes don't have a nucleus, so how might that change protein synthesis? Do you guys have any ideas? Well, yeah. The one way it does change it is essentially, if you remember, what slide are we on? We're on slide 36, so we'll go back all the way to like this slide over here. If this step is transcription and this step is translation, in prokaryotes, transcription and translation can happen simultaneously. In eukaryotes, if this step, or sorry, before the processing stage, if this step is transcription and this step is translation, transcription and translation can't happen at the same time because the transcript has to be processed before it's ready for translation. So this is a big difference in eukaryotes and prokaryotes. Prokaryotes, they can simultaneously transcribe and translate. Backbone to which ribosomes are attached. Okay, cool. So if you can see here, this is a segment of E. coli chromosome. So you see like this, oh, let me move this out of the way. You see like this line? You can kind of see it. This is essentially the DNA of E. coli. And extending from it are polysomes. I think maybe Miss Law meant to write polyribosomes. So extending off this chromosome would be like the transcript or the mRNA, which is being transcribed, and at the same time, ribosomes, which are these dots, are attached to it, and these are all polyribosomes at the same time. It's pretty cool. Oh, so it's over here. Yeah. Cool. So basically, that's kind of it for translation. So we've covered initiation, elongation, and termination. But after termination, we still only have a, a polypeptide chain. So we're not at proteins just yet. We're pretty close though, because remember that in order to get a protein from a polypeptide chain, your protein has to be functional. But many, many different things can happen after you have your polypeptide chain. I guess we can just call it protein from now until it's finally where it is doing its job. So this is basically a slide that can outline many or all of the different options that a protein can go through from first being created to ending up where it needs to be doing what it's supposed to be doing. So in class, what we did was Miss Law would put this slide up, and then it was up to us looking at this slide to create a flowchart of what protein trafficking is. We're not in a class, so we don't have the ability to do this, but uh, I would recommend on your own time, like maybe trying to figure it out. I guess we're gonna go through it together, so it doesn't, you won't really have that chance, but it should be fine. Let's go through it together. There are two types of ribosomes before we get super into this protein trafficking thing. There are two types of ribosomes. There are free, unbound ribosomes and bound ribosomes. Unbound ribosomes are found in the cytoplasm. Bound ribosomes are found in the cytosolic side of the rough endoplasmic reticulum. Cytosolic side means it's the side facing the cytoplasm of the cell. Cytosol is like the cell environment, and the other side of the rough ER would basically be facing inwards into the lumen of the rough ER. So the different types of ribosomes create different types of proteins. Unbound ribosomes create cytoplasmic or cytosolic proteins. Did I think about this the wrong way? I'm pretty sure. Oh, right. And bound ribosomes create secretory proteins. So these are proteins that are secreted. And these different proteins have different final destinations. Cytoplasmic or cytosolic proteins will end up in the cytosol or in different organelles. Oh, I messed up then. Cytosolic side would be the inside. My bad, guys. I think cytosolic, if you have like the rough ER as its like own thing, and then you have the outside cell, this would be the cytoplasm inside or outside, and then this would be the cytosol inside. Oh, that's terrible writing, but I hope you guys understand. So it ends up inside of the rough ER. And the final destination for these proteins are the plasma membrane, as either transmembrane proteins or integral proteins, or they're secreted from the cells through exocytosis, or they end up in lysosomes. So this is a little flowchart describing all the different options. So it all starts with the location of your ribosome. Is it free or is it bound? And from there, you have basically a fork of paths that your protein will go. Cool. So cytosolic proteins, what are they? Well, they're water soluble, and their folding can be aided by chaperone proteins. So chaperone proteins basically provide the ideal environment for folding, and the folding occurs spontaneously. So the polypeptide chain comes through here, goes into this chaperone protein, and then the chaperone protein is capped, the protein is folded, and then it's kicked out, and it's ready to go. Hydrophilic environment doesn't have water. So I can definitely tell you that if it says hydrophilic environment, then it's like you're right. It does mean like not water loving, but I can't tell you if it's the cytosolic side because this is a protein. So this is not like a cell organelle where you have a specific boundary between cytosolic and cytoplasmic sides. I think this is just the entire thing is happening in the cytosol of an organelle. Let me go over this one more time. I think, is it maybe referring to cytoplasm and cytosol? Give me 1 second, guys. Let me do a quick Google search. Cytosol and cytoplasm. Oh, okay. So, I think so, according to the knowledge of Google, cytosolic is basically referring to the intracellular fluid that is present inside the cells. The cytosol comprises of a lot of water, dissolved ions, large water-soluble molecules, and smaller minute molecules and proteins. But the cytoplasm is just the part of the cell which is contained within the entire cell membrane. So that could mean a lot of different things, I guess. Um, that's a good question. I think we may need Miss Law to answer that question. Creating a hydrophilic environment. So maybe it's because cytosolic proteins are usually water soluble, so that means that they like water, or they want, or they are fine like being in the same environment as water, like they won't repel water. But entering into this chaperone protein creates a hydrophilic environment that allows the protein to fold. I guess this protein can only fold when it's in a hydrophilic environment, but its normal environment is somewhere where it can be dissolved in water. That's my best answer for you. Cytosilic side. Hmm. Yeah, I don't know if that answers your question. And it's interesting. I don't know. I'm not too sure what the difference here between a cytoplasmic or a cytosolic protein would be. This is a good question for Miss Law, and hopefully she'll have an answer for us. Any other questions? All right, let's move on. So, what is a signal sequence? Signal sequences are very important, and they're basically like intracellular postcodes. So all translation begins with free ribosomes. So the free ribosome synthesizes a polypeptide chain, revealing a signal in the N-terminus that will then direct the translation complex to the endoplasmic reticulum, or it won't if there is not a signal sequence. So what is a signal sequence? It's 5 to 30 hydrophobic amino acids, and it's at the N-terminus of your polypeptide chain, and this determines the destination for the polypeptide to the ER. If this is there, then this polypeptide is going to the ER. And this is also known as a signal peptide. Okay, let's take a look here. This is a pretty good diagram. So, if you take a look at this ribosome over here, this like green thing would basically be the signal sequence. You can see here on this ribosome, the polypeptide does have a signal sequence, so it's brought to the ER, where it has many different paths to go. Over here, there is no signal sequence, no light green box, so it's not trafficked to the ER. Does that make sense? This is a little diagram. I think we're going to go over this in more detail on the next slide. So, when you begin translation with an unbound ribosome, as a polypeptide is translated, the N-terminus can reveal a signal sequence. So let's say it does. The N-terminus reveals the signal sequence. So then what happens? A signal recognition particle, or an SRP, binds to this signal sequence and halts translation. So the ribosome stops translating. So then this SRP binds to an SRP receptor on the ER membrane, bringing the entire translation complex with it. And when this happens, the ribosome is docked on the ER membrane. So look over here, the N-terminus has revealed the signal sequence. So what happens? An SRP comes and binds to this signal sequence, preventing the ribosome from translating further. What happens then? This SRP is brought to an SRP receptor, docking the ribosome there with it. Step 4. The signal sequence is then oriented into what is called a translocon, similar to a channel protein. You can think about it similarly. And then the polypeptide is threaded through this translocon into the ER lumen. And then the SRP is released and the translation resumes. So this would be over here. Your polypeptide chain is threaded through this translocon into the lumen of the ER. And then afterwards, something called a signal peptidase cleaves the signal sequence. Translation continues as normal, and then when translation is complete, the polypeptide is released into the ER lumen. Signal peptidase cleaves the signal sequence. So if the ribosome is docked, how can translation continue? So translation is stopped by this SRP, essentially. So when this SRP basically recognizes the signal sequence and attaches to it, the ribosome can't continue to translate. But once this SRP attaches to the SRP receptor, the ribosome docks, and then the polypeptide is threaded through this translocon, there's nothing stopping the ribosome from continuing translating anymore, if that makes sense. So then once the SRP has done its job, the ribosome is docked and the SRP detaches, because as you can see here, it's still attached to the signal sequence here, but in this part of the diagram, it's not attached to the signal sequence anymore. The polypeptide has already been threaded through the translocon, and there's nothing stopping this ribosome from continuing what it was doing before. Does that make sense? It's basically momentarily stopped. Yeah, put on pause. Cool. So then afterwards, polypeptide is threaded through, continues translation, and when it's done, everything else normally happens. Uh, basically the polypeptide is released into the ER lumen, and the translation complex disassembles. Once your polypeptide chain is inside the ER lumen, it goes through something called the endomembrane system. So this is from the ER to the Golgi body, to lysosomes, or the cell membrane, or outside. So let's look at different destinations once it's in the ER lumen. Vesicles containing the polypeptide in the endoplasmic reticulum lumen, they can pinch off and fuse into the Golgi. So if you look here, this is one example. Rough endoplasmic reticulum, these little proteins, they pinch off and attach to the Golgi body. That's this example. Vesicles containing the polypeptide in the lumen of the Golgi can pinch off. So if you look at it over here, it can pinch off towards the lysosome. It can pinch off as a secretory vesicle. And other possible destinations, the vesicles can fuse with the cell membrane for exocytosis, or the vesicles can become lysosomes. So these are the two examples. But there's another thing that can happen with these polypeptides that end up in the ER. There's something called a stop transfer sequence that can be on the nascent polypeptide. The stop transfer sequence anchors the polypeptide into the membrane of the rough endoplasmic reticulum, so that once translation is complete, the polypeptide does not get released into the lumen. So everything we saw before up to this step is the same. Signal sequence is cleaved off, translation is continued as normal, but now there is something called the stop transfer sequence that has been translated. If there's a stop transfer sequence, it will basically get anchored. I don't know the mechanism of anchoring this stop transfer sequence into the membrane, but it will be anchored such that once translation is complete, this polypeptide won't just be released into the ER lumen, it'll stick around stuck in the membrane or anchored into the membrane. For a polypeptide or a protein anchored in the membrane like this, there are different destinations for it. Let's look at them. Vesicles can pinch off from the ER and fuse onto the Golgi with the polypeptide still attached to the membrane. And then these vesicles will pinch off from the Golgi body and exocytose to the cell membrane, where the polypeptide becomes incorporated onto the cell surface. So basically receptors. I don't think that this specific example is present in this diagram, but I'm pretty sure this part is not too hard to understand. Cool. So for bound ribosomes, there's so many different paths that you can take. Basically, there's like 3 or 4. And if you've noticed, in almost every example, the protein that ends up at the rough endoplasmic reticulum will be pinched off and sent to the Golgi body, because the Golgi body has a very important role. In the Golgi body, something called post-translational modifications happen. So after translation, the polypeptide chain can be modified, or the protein can be modified in a way that's important to achieving its function. So 4 things can happen: addition, removal, polymerization, or folding can happen in the Golgi body. Let's look at an example. Insulin. So the polypeptide chain of insulin, also called preproinsulin, is not active. So if you just had this stuff in your body, it would not be doing the function of actual insulin. Many different post-translational modifications need to happen before insulin, preproinsulin, is in the active form, insulin. So if you can look here, it looks like this part of the polypeptide is just cleaved off, and afterwards it must create like there's like some bonds that happen, but essentially this is not equal to this until post-translational modifications have happened. Very important. Here's some extra information. Types of RNA. This is good to know. I think we were tested just briefly. I think it was like 1 or 2 multiple choice questions on this. But as you study all the other PowerPoints, this stuff will basically come because you need to understand this to understand all the concept we learned before. Here's a little comparison of transcription and translation. Take a look at it. Here are some homework questions. These are some actually good questions to do. Does anybody have any questions about protein trafficking or post-translational modifications? I'm sorry about the confusion with cytoplasm and cytosol. Hopefully Ms. Law can straighten that out for us. I'm a little bit confused myself. Other than that, does anybody have some questions? No questions? Okay, cool. That's it.